Good morning students. Today in this session we are going to discuss about the packaging of DNA helix. So, packaging means what here? Actually the DNA is a thread like structure, elongated structure. So, how it is packed in the simple very small means it is microscopic. So, we are referring here microscopic nuclei. So, the nucleus of a cell, understood. So, in that very small structure the DNA is present, understood. So, how it is packed? So, all we are going to discuss here. Before going to discuss how it is packed and what content it has. So, let us we see once again the Watson and Crick model in brief. Okay. See, this is the Watson and Crick model, which is double stranded DNA structure, it is. Okay. So, this is what understood. So, these are the two strands. Here you can mention the strands. This is the puc prime. Okay. So, these are the two strands. So, there is a inside the runs what we called the nitrogen bases. So, there is the nitrogen bases. So, this structure already we have seen in previous video. So, now, so how to identify the length of DNA, whole length of DNA and how much elongated it is, and so how it is packed. So, to understand that, so we should know the some parameters regarding this structure. So, in between two nitrogen base pairs, the length or the distance you can consider either length or distance between two consecutive or adjacent, you can consider it as adjacent. Two adjacent means these two, okay, neighboring, okay, two consecutive nitrogen base pairs, I commonly we can use it as a base pairs, BP. Okay. So, what is the value? The length or the distance between two consecutive nitrogen base pairs. So, already we know that for a complete turn of a DNA, so how much it is? 34 angstrom in it, 34 angstrom, understood. So, this 34 angstrom is the for complete turn, and in that one complete turn there are 10 base pairs, because in Watson and Crick model, so B form of DNA it is right handedly coiled structure it is, okay, clockwise direction it will turn. So, in complete turn there are 10 nitrogen base pairs are there, 10 nitrogen base pairs. So, one nitrogen base pair, so sorry from one to another. So, this is what we call consecutive base pair or adjacent base pair, you can consider adjacent or neighboring base pair, understood. So, this distance is, it can be calculated because here 10 is there, okay. So, for 34 angstrom 10 base pairs are there, means it will be approximately 3.4 angstrom, okay. So, that is the length or the distance between two nitrogen base pair is equal to, how much it is? 3.4 angstrom unit, 3.4 angstrom unit. So, usually angstrom unit can be converted in either nanometer, 0.34 nanometer, or even you can consider it is in meter, 3.34 into 10 raised to minus 9 meter. Yes, 0.34, this is about the 0.34 into 10 raised to 9 minus 9, 10 raised to minus 9 meter. So, this when you understood, so then we can calculate the length based on the number of nitrogen bases. Okay. Now, see, it can be calculated, the length, the total length can be calculated, total length is calculated, it can be calculated, the total length of DNA, total length of the whole DNA of a typical cell. So, the total length of DNA in a typical mammalian cell, can be calculated, How it is calculated? So, we should know the data. So, already you know this one, the base pair in between two base pairs. So, there is a 0.34 into 10 raised to minus 9 meter length will be there, and once you know the number of base pairs, when in a question, so they may give you number of base pairs or they may give you the total length only. Okay, by that we can calculate the, so whether length or even the any out of these three datas here. So, one is in between two consecutive nitrogen base pairs, or in that question may have either the total length they will give. Okay, let us see later. So, how can it will be calculated? Simple one formula we can make here. So, the total length, the length of DNA is equal to, the length of DNA is equal to number of nitrogen base pairs, multiplied with its the distance of two consecutive BPs. Okay. So, by this formula we can calculate either the length or number of nitrogen base pairs, or it can be also calculated this one. Out of the three important datas are here. Okay. Even in NEET level some questions may be framed on this simple formula. Okay. So, this is the total length is nothing but it is the multiplication. How can you define it? It is a multiplication of the distance between two consecutive BPs and the total number of nitrogen base pairs. This is also number means what? Total number of nitrogen base pairs. Total number of nitrogen base pairs. For example, elongated chain is here. Okay, elongated chain, how many turns are there, and in each turn how many nitrogen base pairs are there. On that basis we are going to discuss. Of course, the human DNA is a B form DNA, which is usually in almost all naturally. So, the B form DNA is there. That means 3.4 will be the distance between two nitrogen base pairs. Understood? So, that is why. So, here, so you can how can you identify the typical cell DNA in a mammalian cell? I have taken here mammalian cell. So, it has the nitrogen base pairs, total number of nitrogen base pairs. So, let us we can calculate here 6.6 into 10 raised to 9 BPs. 6.6 into 10 raised to 9 BP. BP means here base pairs. So, total number of nitrogen base pairs in a typical cell, mammalian typical cell, a model cell we have taken here. Okay. So, that DNA shows this much of base pairs. This much of base pairs are present here. So, already we know that the 3.4 angstrom, which we have converted for calculation purpose, we should notice this one. Okay, we should take this meters in meters. 3.4 into 10 raised to minus 9 meter, 10 raised to 10 raised to 0.34 into 10 raised to minus 9 meter. So, when we calculate this, when we calculate both, so simply you can apply the mathematics. So, these will get cancelled and it is approximately to, so its value will become, when you multiply this 0.34 into 6.6, so you will get the value 2.244. Okay. So, it will be, it is a number, so we can apply this meter. Okay. The length is there here, so we can apply the meter. Next, approximately 2.2 meter is the total length. Yes, in a typical mammalian cell, the DNA length is about 2.2 meter. Understood? So, in this way we can calculate the length. Okay. So, now one more, if they will give us the length, we can also calculate the total number of nitrogen base pairs. Okay. So, in E. coli bacteria, so you can take that also. E. coli. E. coli is the, as you know, Eustachian coli is the bacteria which is found in the colon. Okay. Digestive tract, that is colon means digestive tract, large intestine part it is. So, where E. coli bacteria will be there. So, in this bacteria, as you know, bacteria it is a prokaryote, okay, because there is no nucleus. So, you should remember, when you consider this is the bacteria, the DNA present in bacteria or prokaryotes is different rather than this eukaryotes. Understood? So, in eukaryotes there is a nucleus. See, this is the what you call the cell, and here there is a nucleus. So, in that chromosomes, what we call the chromatin material, and so we know that the simple cell structure. Okay. See, this is what the DNA, double stranded DNA, which is there in this thread like structure. Okay. So, the two, how much its length? The mammalian, if you consider it is a mammalian cell, so about 2 meter length is there. And what is the dimension or diameter of this nucleus, whole nucleus? So, very small it is. Okay, microscopic, not meters together. Cell is a microscopic. Okay, you can say meter. So, it is a long thread like structure which is packed in the simple, very minute what you call the nucleus. In that nucleus, which is compactly there inside the nucleoplasm. That means that is the liquid substance present in the, that is nucleoplasm. In this nucleoplasm there is a chromatin. So, chromatin is formed by DNA only. DNA and proteins are there. In the next we will discuss the details. Chromatin means it is a DNA plus protein. So, both together we call it as chromatin. Later we will discuss that details. That is what the packaging of DNA helix, how it is packed. 2.2 meter length DNA thread and with proteins, how it is packed, that we are going to discuss after this one more calculation. So, in E. coli bacteria, the correct form of nucleus is not there. So, instead of that, there is a what is there? Yes, there is a large looped structure, double stranded structure, which we observed that like a loop. So, in the center of this one, which is not scattered. Okay. In prokaryotes, the prokaryotes means single celled, of course. Okay, single celled. This is the bacteria when you consider, this is cytoplasm, and in the center of this cytoplasm there is a chromatin or the material is there, DNA material, chromatin material is there, DNA is there, but it is not scattered throughout the cell. Whatever the area of the cell is there, no, which is not scattered. Understood? So, which is present at the center and it is in a loop like manner, loops we can say. Okay. So, such a type of what you call the nucleus is called nucleoid. What you call? Nucleoid. We call it as a nucleoid. Understood? Which is not a nucleus, because the membrane is not there. Here membrane is present. Here what we call this chromatin material is covered by a membrane, complete membrane is there. Here there is no membrane, membrane is absent due to absence of membrane. So, primitive type of nucleus we can say. So, in this the nucleoid we call. Nucleoid means it is a DNA and protein. Understood? DNA, nucleoid, it is containing DNA plus protein only, that is histone proteins will be there. Okay. Now, so what is the question here? They are giving us the length of this E. coli, what you call the length of E. coli DNA. The length of E. coli DNA. The length of E. coli DNA is how much? So, that is they are given as a data 1.36. How much it is? 1.36 mm. They are giving us in mm. Remember, either angstrom unit or in any unit they can give you, okay, in the question. So, we should identify that 1.36 mm is given. So, in that 1.36 mm, they are asking you calculate the number of base pairs. What they are asking? Calculate the number of base pairs present in it. Understood? Present in E. coli DNA. Okay. So, the length is given, length of that DNA is how much? 1.36 mm, and they are asking you to calculate number of base pairs. What is the number of base pairs? Go through that simple formula. So, the length of total DNA, total length of DNA we can consider is equal to total number of base pairs multiplied by the distance of two base pairs. Understood? So, more I am simplifying here. Okay, total T means here total number of base pairs, into distance of two consecutive base pairs. So, this is what the even you can give the definition also. Okay, by this formula, if you remember this much, okay, you can also define the total length of DNA. Total length of DNA is nothing but it is the multiplication of the total number of base pairs present in it and the distance of two consecutive base pairs or adjacent base pairs. Understood? See here, there is a what you call the data is here. Yes, the data, how much is given? The length is given already. So, you can convert it into meters. Okay. So, millimeter is there here. So, you can I will take directly here 1.36 mm is given. I will put the data. So, here we do not know, we have to calculate number of base pairs here, total number of base pairs we do not know. Okay. So, this we have to calculate. So, we can take it as x multiplied by the distance of two base pairs is already, that is what B form DNA if it is there, we can consider this is the length, that is 0.34 into 10 raised to minus 9 meter, which is in meter and it is in mm. So, that so you can take it as you can convert it. So, for example, here we can convert it by meter, okay, from mm to meter, that is 1.36 into 10 raised to minus 3 meter is equal to x into 0.34 into 10 raised to minus, yes, this is what the data is there. So, we have to identify the x value here, x value. So, now this is become x is equal to so 1.36 into 10 raised to minus 3 divided by 0.34 into 10 raised to minus 9. So, that is in both in meters now. Okay. So, now we can simply calculate this one. So, when you calculate this, so now we can, after calculation, so by simple mathematics we can apply and calculate it. So, finally, we will get the answer 4 into 10 raised to 6, 4 into 10 raised to 4 into 10 raised to, that's approximately 4 into 10 raised to 6 base pairs. So, when you go to this one, so another step also you can take 1.36 into 10 raised to minus 3. So, if it is become plus 9 divided by 0.34. So, another step also you can write. So, now it is become value is become 6, understood. So, in this way, so when you multiply this, so there is a answer will become 4. Okay. So, now you can calculate the and you will get the answer. This is what the answer of x. So, now the total number of base pairs are 4 into 10 raised to 6, 4 into 10 raised to 6 number of base pairs are present in this much of length 1.3 mm, 1.36 mm meter length in E. coli, okay, in E. coli bacteria. So, this is what the about the length or, so in a NEET level such questions will arise or framed. So, for that this simple formula can help you. Okay. Dear students, in the next video, the packaging of DNA helix we can discuss. Okay. Please subscribe the channel.